I'm going to tell you today a little bit about the basic biology of autophagy before moving on to describe why we think this is an important process in the context of neurodegenerative diseases. Autophagy was classically thought to start where cells form this double-layered cup-shaped structure called a phagophore in the cytoplasm to engulf a portion of the cytoplasm. And the engulfment occurs after the edges of this cup-shaped structure extend and then fuse to form the autophagosome. The autophagosomes are formed more or less randomly throughout the cytoplasm of most mammalian cells, and then are trafficked via dynein motors along microtubules to the microtubule organizing center of the cells where the lysosomes are clustered. And this enables fusion with the lysosomes to subsequently occur. And after the fusion occurs, the hydrolases in the lysosomes degrade the contents of the autophagosomes. One of the long-standing mysteries in the field is where the membrane that contributes to autophagosomes actually comes from. And there have been suggestions that under a situation where autophagy is induced above basal levels, that some of the membrane might come from the endoplasmic reticulum or from mitochondria, for instance. And our data suggests that an important source of membrane is the plasma membrane. And this gives rise to autophagosome precursor structures that have single membranes that then undergo a series of homotypic fusion events to create this double membrane structure. Autophagy is regulated by many different signals in the cell. One key signal, in fact it's a primordial signal, that's conserved from yeast to man, is mediated by the target of rapamycin protein that we've already heard quite a lot about today. And the M just stands for mammalian. And this protein, it's a kinase, serves as a negative regulator of autophagy, and one can induce autophagy by inhibiting the target of rapamycin with drugs like rapamycin. There are a number of other ways that you can induce autophagy through this pathway. One is when you starve cells, you inhibit the target of rapamycin. But there are other ways one can do this as well. So recently we found that one way of doing this is by moving lysosomes around in the cells. Because most of the target rapamycin complex that regulates autophagy is very closely associated with the lysosomes. So we found that if one clusters the lysosomes very tightly around the microtubule organizing center, one decreases the activity of the target rapamycin complex and thereby induces autophagy. And one does this by increasing the rate of formation of autophagosomes. But in order for autophagy to work properly, you don't only have to increase the rate of formation of autophagosomes, you've also got to increase the rate of delivery and fusion of the lysosomes. And this is one mechanism where one can do this, because by placing more lysosomes in this position, one places more of them in the incoming travel path of the autophagosomes and thereby enables more efficient autophagosome lysosome fusion as well as increased autophagosome formation. The reason why we think this occurs in terms of the induction of autophagy by decreasing mTOR activity when you move the lysosome very closely to the microtubule organizing center is that you're taking them further from the plasma membrane where the upstream activators like AKT are situated. You get the converse effect, of course, if you drive the lysosomes with the accompanying target rapamycin complex to the plasma membrane, and when you do this, you increase the activity of the target rapamycin complex and you switch it for autophagosome formation. There are signals that regulate autophagy that are independent of the target of rapamycin, and I'm going to just describe one of these because it becomes relevant for one of the examples I'm going to give you towards the end of the talk. And this is a process that's regulated by a key autophagy protein called Beclin-1. Beclin-1, under basal conditions where autophagy proceeds at a slow rate, is found in a significant pool. So most of the Beclin-1 is found associated with microtubules through an interaction with this protein called BIM. And this takes Beclin and its associated protein, BPS34, away from the endoplasmic reticulum, where it needs to do its job to most efficiently upregulate autophagy. We found that one could induce autophagy through this pathway through starvation or by modulating the activity of various kinases, which phosphorylate them. And when you phosphorylate, when you break this interaction of BIM and Beclin-1, and you enable the Beclin-1 complex to move to the endoplasmic reticulum to activate autophagy. The reason we're interested in autophagy is not primarily for its own sake, but because my lab has a major focus on neurodegenerative diseases. And most of the adult onset neurodegenerative diseases, and indeed most of the childhood onset diseases that I'll come to later, are associated with the formation of inclusions in the cytoplasm of cells. You see this with tau in Alzheimer's disease, you see this with Huntington's disease, adult onset Huntington's disease, you see this with alpha-synuclein in the Lewy bodies in Parkinson's disease, as well as in the other examples in slide. When these diseases are caused by autosomal dominant mutations, very frequently the autosomal dominant mutation does two things. It makes the protein aggregate protein, but it also changes this protein into a poison for the cell. And so the strategy we've taken in the lab is to see if we can find ways of enhancing the removal of the poison for the cells and not really do much to the clearance rate of the wild-type protein. And about 10 years ago, we found that this could be modulated by the autophagy lysosome pathway. We found that if we decrease the rate of autophagosome formation or decrease the rate of autophagosome lysosome fusion, you slowed the clearance of the precursors of the aggregates that you see in Huntington's disease, for example. That results in two things. One, it increases the levels of both the soluble and the aggregated species of the toxic protein, and as a consequence, it increases the toxicity of the protein in the cell or even in vivo systems that we study. Perhaps of greater interest, we found that if we increase the rate of autophagy, we enhance the clearance of these mutant proteins in cells, in Drosophila and in mice, and we reduce the accumulation of the toxic proteins and alleviate the toxicity. It's important to point two things out. The first is that the dependency of the mutant aggregate proteins for autophagy is pretty high, while the dependency of the wild-type counterparts is pretty low. So if you decrease the activity of the autophagy pathway, you hardly impact on the clearance of the wild-type protein at all. And I can discuss the reasons for that in the questions. The second point is that our initial experiments were done with rapamycins. And the reason we used rapamycins is that 10, 11 years ago, when we started this line of investigation, these were the only drugs that were used in man that were known to induce autophagy. And it's important to stress that rapamycins are designed for long-term clinical use in people, and one of the initial indications was as an immunosuppressant for people that had kidney transplants. So that brings me to one other point. The experts in the field on the target of rapamycin will know that it affects many key processes in the cell. And I just want to stress the point that we've gone on to show in a variety of systems, and others have replicated that at least in Drosophila models where you can test this in vivo, the effects of the rapamycin on autophagy is dependent. And I can explain the experimental paradigms later. So we were very excited. We had a sort of broad concept, and we have proof of principle not only in cell-based systems but also in a variety of animal systems. And so these were experiments. With Huntington's disease, we wanted to see whether this could be extended to other diseases in the category that I showed in the slide of neurodegenerative diseases. So we started looking. Huntington's disease is one of 10 diseases known to be caused by abnormally long polyglutamine tracts in the protein. The expansion of the polyglutamine tracts constitutes the mutation. And in some of these, the mutation causes a protein to aggregate in the cytoplasm and some in the nucleus. And it's the cytoplasmic diseases we're interested in. So we've taken ataxin 3 as one of the examples of the disease where, in fact, the protein has some residence in the cytoplasm. It's the most common spinocerebellar ataxia, and we showed this as a very good autophagy substrate. We did experiments with isolated polyglutamine expansions, and we showed that they're good autophagy substrates in cells, and their toxicity in Drosophila models could be ameliorated by upregulation of autophagy. Likewise with long polyalanine repeats, which are associated with 9 diseases. Perhaps of greater relevance, we showed that wild-type tau, which is associated with Alzheimer's disease, and indeed it's thought to be a major mediator of toxicity in Alzheimer's disease, we showed that this is an autophagy substrate, and that its toxicity in Drosophila models could be ameliorated by autophagy upregulation, and we showed the same with point mutations in tau that give you frontotemporal dementia. Finally, we showed that mutant forms of alpha-synuclein that give you autosomal dominant forms of Parkinson's disease are very good autophagy substrates. So the next thing we were planning to do was a whole lot of experiments in mice, and so Fiona Menzies in the lab did an experiment in a spinocerebellar ataxia type 3 mouse model, and she showed that we could ameliorate toxicity even after the onset of signs in this mouse model. which was quite gratifying, and almost normalized the phenotype in this very slowly progressive model of the disease. We were planning on doing a whole lot of other mouse models, and we're delighted to see that other people have done these experiments, which are rather time-consuming. So what the slide shows are data from mouse models of all these diseases shown in green. So it's only mouse data, where people are followed on the same type of idea and tested whether autophagy upregulation can be beneficial in such mouse models, and I put the slides because, these examples on the slides because the answers are yes. So that was very exciting for us. When we're thinking of treating these type of diseases, one class of diseases that might be particularly amenable to such therapy are autosomal dominant diseases like Huntington's disease, where you can identify people who are at risk long before the disease starts to manifest. And so you could potentially treat them when they're still well and try to delay the onset of disease. Now, although rapamycin is widely used, it's not like taking half an aspirin a day. So we wanted to see if we could find safe and possibly more specific drugs to modulate this pathway. And so we've taken a variety of approaches. One is to take target compounds, or from, in this case, data that we generated in the lab ourselves. The other is to do genetic screens. So I've shown this slide because it represents the first characterized mTOR-independent autophagy regulatory pathway. This shows a cartoon of one of the pathways that regulates the target of rapamycin with upstream insulin signaling, and showing that rapamycin blocks this pathway to induce autophagy. What Soba and Saka found in the lab was that another way of regulating autophagy is by decreasing the levels of inositol triphosphate. This acts independently of the target of rapamycin pathway and additively to its pathway, and perhaps most excitingly, can be regulated by 3 drugs at least that are in clinical use for treating brain disorders chronically. That's carbamazepine, valproate, and lithium chloride. As I said, we've also undertaken a number of screens for autophagy modulators, and I'm just going to focus on the more recent published screen where we focused on drugs that had already been used for other purposes and tested whether they're regulating autophagy. And we identified a series of compounds that regulated autophagy independently of the target of rapamycin, that enhanced the clearance of toxic proteins like mutant huntingtin and mediated protection in cell, Drosophila, and zebrafish models of Huntington's disease. I'm not going to go through all the signaling. The drugs that do the trick are shown on the slide, and again, in the question time, I'm happy to describe how they might be connected. But I just want you to focus on this compound ruminidine, which induces autophagy by binding to imidazoline receptors and decreases cyclic AMP in order to mediate its autophagy-inducing effects. And I want to focus on ruminidine because it's a centrally acting antihypertensive drug. By that I mean it's a drug that's designed for treating people with high blood pressure, and so the people will be taking the drug for very long periods of time. And it reduces blood pressure by acting on receptors in the brain. And it acts on the same target for blood pressure as it does for autophagy. And these receptors are very widely expressed throughout the brain in the regions which will be affected by many of these neurodegenerative diseases. Perhaps of greatest appeal to me was the fact that this is a very safe drug. So the side effect profile in individuals taking the drug is the same as for hypertension. The conventional dose is the same as people taking placebo. We went on to show that it induces autophagy in primary striatal neurons, as we saw in all the other cell systems that we looked at. And we looked into primary striatal neurons because this is a cell population that's most susceptible to the Huntington's mutation. We showed that it reduces mutant huntingtin levels in the mouse model of Huntington's disease and alleviates signs of this disease to a very similar extent to which we saw with rapamycin. And so we're currently in the midst of a safety trial with Roger Barker in Cambridge to test this drug in early stage Huntington's patients to ensure that there are no unforeseen drug gene effects in the Huntington's patients. And hopefully this is a box we can tick as further armamentarium, increase our armamentarium to try to justify a proof of concept study in early Huntington's patients. I'm going to end my talk by describing what happens when neuronal autophagy is blocked. This concept was introduced by 2 Japanese groups led by Noboru Mizushima and Masaki Komatsu. And what they did were conditional genetic knockouts of two different autophagy genes in mouse neurons. So they basically just removing the autophagy genes in mouse neurons, and they got the same data, fortunately. They got two primary findings. First is when they stained for ubiquitin, they found that the autophagy defective neurons had the appearance of these multiple ubiquitinated aggregates. The second is that they found decreased cell numbers, decreased neuronal numbers in the autophagy knockout brains. And so you can see this is the Purkinje cell layer of the cerebellum of a normal mouse. You can see fewer cells in the autophagy defective neurons. This is associated with increased tunnel staining, suggesting that there's increased cell death leading to this defect in neurons. There were two additional findings that I'd like to focus on. One wasn't too unexpected because they saw the accumulation of this protein called P62, which is a ubiquitin binding protein that previously had been described as an autophagy substrate. So the fact that it's also occurring in the mouse model is no great surprise, but I want to stress it because it becomes important later in my talk. What was quite interesting is when they did immunohistochemistry, they saw that the P62 staining overlapped almost perfectly with ubiquitin staining. I think these studies from the two groups were very important because they set the scene for trying to understand what might be going wrong in various neurodegenerative diseases, where we now know there are multiple conditions where there are defects in different stages of the autophagy pathway. And so I'll give you some examples. And the color code is meant to show defects at different stages of the pathway in different diseases. So the blue diseases are conditions where there's a defect in autophagosome formation. The Fora disease is a degenerative disease which leads to epilepsy and very early death. And in collaboration with Bernet's group, we showed that this disease has defective autophagy both in cell-based systems and in mice. And the reason for this is that the mutation basically hyperactivates mTOR. Perhaps more interesting or broader significance are data in the context of Parkinson's disease. The protein that accumulates in Parkinson's disease aggregates is called alpha-synuclein. Excess alpha-synuclein is sufficient to give people Parkinson's disease because they're rare individuals who have duplications of the alpha-synuclein gene locus, and they get a familial form of early onset Parkinson's disease. Ashley Winslow in the lab showed that if you've got too much alpha-synuclein in cells, and with others in Drosophila and mice, she showed that if you've got too much alpha-synuclein, you block autophagosome formation. Indeed, if you take an alpha-synuclein transgenic mouse, for instance, which has no phenotype on its own, and you cross that to a Huntington's mice, we found that you bring the phenotype on prematurely in the Huntington's mice when you've got excess alpha-synuclein. What was additionally interesting is that we found if we reduce the levels of alpha-synuclein, then we induce autophagy. And again, we've got data in cells, Drosophila, and mice. So if you take a Huntington's mouse and you put it on an alpha-synuclein defective or alpha-synuclein null genetic background, you induce autophagy in the brains of these mice, you enhance the clearance of the mutant protein, and you exacerbate the phenotype to some extent. So this suggests that variations in the level of alpha-synuclein around the physiological level modulate autophagy. In the first slide, I explained that in order for the autophagosomes to be able to fuse with lysosomes, they need to be trafficked by these dying motors. And we show this, again, in cells, Drosophila, and mice. And we know that there are defects in the machinery in forms of motor neuron disease and in a rare form of Parkinsonism called Perry syndrome. These diseases, where there's defects of dynan activity that I mentioned, have aggregates and have neurodegeneration, and I think it's quite plausible that the defect in autophagy that is seen in this situation is contributing substantially to the phenotype. Finally, in order for autophagy to work, the lysosomes need to function. In collaboration with Andrea Balabio's lab, we showed that in lysosomal storage disease, which are the most common neurodegenerative diseases of childhood, there is defective autophagosome lysosome fusion. And this leads to the idea that in lysosomal storage disease, the toxicity is not simply mediated by the accumulation of the substrates of the particular hydrolases that might be defective, but there's an additional toxic insult in that one gets impaired clearance of all autophagic substrates. This concept has been extended in a way by others So Randy Nixon has shown that in Alzheimer's disease caused by presenilin 1 mutation, there's a similar type of defect at the level of the lysosome, and in rare forms of frontotemporal dementia caused by mutations in the escort complex, and Simonson and others have again shown that these defects are autophagocyte lysosome fusion. Finally, there's an additional concept that's been introduced by Ana Maria Cuervo looking at cell models of Huntington's disease, where she's shown that the mechanistic procedure of autophagy is intact. However, there's a defect in cargo loading in some of the autophagosomes because she thinks that the mutant Huntington is so well incorporated into the autophagosomes that it shields the accessibility of certain other substrates like mitochondria. In addition to genetic lesions, which cause a defect in autophagosome formation or in autophagosome lysosome fusion, there are also environmental insults associated with neurodegeneration that contribute. So one of the features of Alzheimer's disease is excess nitric oxide. And so Vansarkar in the lab showed that nitric oxide blocks autophagy, and it does this through a double whammy mechanism. So if you remember the two signaling pathways I showed you earlier, it inhibits both of those signaling pathways. So by nitrosylating IKK beta, it results in mTOR hyperactivation, and by nitrosylating JNK1, you decrease the activity of hyperactivity. So we wanted to know what happens if you decrease nitric oxide formation in models of neurodegenerative diseases. And so we did experiments in cell-based systems where we had reduction of autophagy and protection. We did experiments in Drosophila, we had them genetically, and we had similar benefits. And I'm just going to end up by showing you some data in our zebrafish model of Huntington's disease. This model expresses the mutant Huntington in the photoreceptors in the rods of the zebrafish. And the mutant Huntington, but not the wild type form, is these very clear aggregates, and they're easy to spot because the mutant Huntington is GFP2 active. If you treat with rapamycin to induce autophagy, you get few of these aggregates, particularly focus on the stoppers of the retina. And likewise, if you decrease nitric oxide formation with this drug L-name. We went on to show that the benefits of L-name in the system were dependent on normal lysosomal function, because if you poison the lysosomes with ammonium chloride, we get no benefit of the L-name in the system, so it's not acting predominantly through some other mechanism. So finally, I just want to introduce the concept of how autophagy blockade might manifest with toxicity. And it might do this through multiple pathways in the cell, but one of the pathways that is affected is the ubiquitin proteasome system, which is one of the other key pathways that clears intracellular proteins in combination with autophagy. So here is a cartoon of an autophagosome degrading P62, which is the protein that accumulates in the lysosomal storage diseases, many of the neurodegenerative diseases, and the autophagy domains. And so P62 is broken down by autophagy. This is a cartoon of a ribosome making a new protein in purple that gets ubiquitinated. So the ubiquitin is a small protein that gets covalently attached to exposed residues on this purple protein by a series of enzymes. After the initial ubiquitination, further ubiquitins are added, so you form a ladder of 4 or more ubiquitins that serves as a recognition signal for transport of these proteins to the proteasome where they can get degraded. One of the proteins that mediates subsequent attachment of ubiquitins, and in some cases delivery of such proteins to the proteasome, is a protein called P97, which binds ubiquitin-mediated proteins as well. Now, parenthetically, it's worth mentioning that both P62 and P97 are mutated in neurodegenerative diseases that manifest neurodegeneration and aggregate formation. So we found that when we blocked autophagy, we slowed flux in the ubiquitin proteasome system. And we did this not by affecting the activity of isolated proteasomes, or by obviously affecting ubiquitination. We think we impaired the delivery of such ubiquitinated proteins to the proteasome. And this is what we think is going on. You accumulate P62 when you block autophagy. And this is necessary and sufficient to mediate the effects. So if you knock down the expression of P62, you don't block floods or slow floods in the proteasome system when you impair autophagy. So it's necessary, it's sufficient, because if you simply overexpress P62 in normal cells, you slow flux the ubiquitin proteasome system. It's dependent on the ubiquitin binding domains of P62, and we could show that by mutating those domains of P62. And it appears to be mediated mediating its toxicity by competing with P97 for binding to ubiquitinated proteins, because she showed biochemically that they compete in this situation, and that if you overexpress P97, then you can rescue the effect of P62 on the ubiquitin proteasome system. So this has two important consequences. If you slow flux through the ubiquitin proteasome system by blocking autophagy, then you accumulate substrates of the ubiquitin proteasome system. One of the key roles of the ubiquitin proteasome system is to regulate the levels of short-lived proteins that are critical regulators in the cell. One of these proteins is P53, which we demonstrated there was an authentic ubiquitin proteasome substrate and wasn't cleared by autophagy. However, P53 accumulates when you slow down autophagy to very great levels, both in cell-based systems and in mouse models. And we think it accumulates to levels that would be sufficient to enhance susceptibility to various toxicity in cells. The other consequence is that P62 accumulates, and we think that this is serving as a seed for those aggregates I described, because the P62 tends to oligomerize, and therefore would aggregate by itself, and it would suck in ubiquitinated proteins because it has ubiquitin binding domains. So I've told you quite a lot in 20 minutes, but I'd like to leave you with two more words. The first is that autophagy upregulation might have a variety of benefits in a range of neurodegenerative diseases. I showed you data explaining how the autophagy upregulation enhances the clearance of various toxic cytoplasmic aggregate proteins. I didn't mention, but I think it's relevant, that autophagy upregulation also protects against apoptotic insults and does so by reducing the rate of caspase activation. And this is important in neurodegenerative diseases because you can get subapoptotic levels of caspase activation, which could mediate toxicity as well. We and subsequently others have shown that this strategy has protective benefits in a range of different neurodegenerative disease models, and this can be mediated by drugs that act on the target of rapamycin, but also on mTOR-independent pathways. The second part of my talk hopefully left you with the idea that autophagy compromise is a feature of many neurodegenerative diseases as well. And the consequences of this would be to enhance the susceptibility to cell stress and to increase aggregate formation in the cell. And the last data slide I showed was to give you the flavor of the idea that it might not be simply the autophagy compromise that is directly mediating this toxicity, but might be due to knock-on effects on a variety of cellular homeostatic processes, including the ubiquitin proteasome system. So you have impaired protein clearance through the two main pathways in the cell. Finally, this was work done over many years by many people. I've shown the main people in my lab on the various data slides. I just want to acknowledge some key collaborators. We've collaborated over many years with Steve Brown in Harwell, and his group with Abraham Acevedo were involved in a study of the alpha-synuclein mice as well as the dynan experiments. Kihiro Kane has been a long-standing partner in crime in all the Drosophila experiments I mentioned. Andres Floats has collaborated with us in writing signaling experiments, like risk gave us the spinal cerebellar attacks in type 3 mice before they were published. The first chemical screening we did was in collaboration with Stu Schreiber's lab. Zebrafish model was made by Paul Cullen, who was a university postdoc that was seconded to a biotech company that used to be in Cambridge, and now we do the zebrafish work in-house in Cambridge. And Andrea Balladio collaborated with us in the lysosomal storage disease studies that I mentioned. And of course, I need to acknowledge the generous funding that we've had without which this work would be possible. Thank you very much.
Okay, so we have